Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing PUMDET question paper for the subject Life Science conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Life Science PUMDET being solved, you can go to my channel Omni Bioentrance. In there you see for the playlist. In the playlist you see for PUMDET and there you will find. So first question, flagship species, the most abundant and cared species in a region. That is not the correct answer. Threatened species selected to act as an icon or symbol for a defined habitat issue or environmental cause. So that is the correct answer. That means the flagship species, they are iconic species, and they represent a particular environmental issue or a campaign, and they are often used to raise awareness to support conservation efforts. So that is flagship species. A point mutation that replaces a purine with another purine or a pyrimidine with another pyrimidine is a transition mutation. In transition mutation, purine is replaced by another purine and or a pyrimidine is replaced by another pyrimidine. Then nonsense mutation, it is a type of point mutation and it results in premature stop codon. In this type of mutation, the codon is changed to a stop codon. That is why you get premature or a truncated protein. Okay. Then silent mutation, it does not change the amino acid after mutation. That is, if it was coding for alanine, then after mutation also it will code for alanine itself. That is silent mutation. Transversion means the version is changed. Remember like that between transition and transversions. That is, version is changed. That is, purine is changed to a pyrimidine or a pyrimidine is changed to a purine. Version is changed. So that is transversion. You can remember it that way. Then next one, the bacterial genus where sterols are present in the cell membrane. So it is in the case of mycoplasma where sterols such as cholesterol is present in the cell membrane. Then which of the following resistance mechanism describe the function of beta lactamase? So it is drug inactivation. Beta lactamase, it is an enzyme that inactivates the beta lactam antibiotics. And your beta lactam antibiotics, they are penicillins. So these beta lactamases, they will hydrolyze the beta lactam ring and it renders the antibiotic ineffective against bacteria. Efflux pump, it is a protein that removes antibiotic from the bacterial cell. Target mimicry means here the bacterial protein, they will mimic the target of the antibiotic. So the antibiotic, it will not bind to the target, it will bind to this bacterial protein which is mimicking the target. So it will not cause any effect to the bacteria. Target overproduction means the bacterial cell will produce excess amount of the target protein. Target protein means the one with the protein that is targeted by the antibiotic. So in excess, that protein will be produced, and this will result in reduction in the effectiveness of the antibiotic. So here they have asked function of beta lactamase. It is an enzyme and it is involved in drug inactivation. I told you how. Then coliforms are used as indicator organism of domestic sewage pollution because they are pathogens. No, they ferment lactose. No, they are abundant in human intestine. That is correct. They are abundant in the human intestine and are present in high numbers also in the sewage. Then identity between two antigens can be verified by. So it is by Octerlony double immune diffusion that you can identify two antigens. I will explain. If the antigens present in both the wells of the Octerlony plate, this is the plate, these are the antigens, small a, this is capital A, which is antibody. So if the antigen present in both the wells are the same, then you will get a curve of pattern identity. That is like this. The curve will be like this. Okay. But if the epitope of antigen in one well is A1, A2 and the epitope of another antigen in the second well is A1, A3, that means A1 is common in both the antigens and A1, A2 is the antibody. The A1, will react with capital A1, and here from this antigen also the A1 will react with capital A1, and you will get a pattern similar to pattern of identity, that is this curve. But since this epitope is different, it is A2 here and A3 here, and the antibody is having A2, it will react with this antigen and it will give rise to a spur. So that is case of pattern of partial identity. And pattern of non-identity means the antigen present in both the wells are entirely different. And the antibody, it will form an independent precipitation line for both the antigens, and the two lines, they will cross each other. That is Octerlony double immunodiffusion. So Octerlony double immunodiffusion, you can identify two antigens. Then rocket immunoelectrophoresis, it is a quantitative technique and it measures the amount of antigen in the sample. Single radial immunodiffusion also, it is a quantitative technique to measure the concentration of antigen in the sample, not identity between two antigens. Then denaturation gel electrophoresis, it is used to separate and analyze proteins, DNA, etc. and molecules separate on the base of their size and charge. So it has nothing to do with identity between two antigens. So that's it. Then an ecotone is, so ecotone, it is a transitional zone between two ecosystems. Okay, and these ecotones, they exhibit higher species diversity than the two ecosystems, and because they contain a mix of species from both the ecosystems as well as certain species that are unique to this transitional zone. So the answer here is higher species diversity compared to adjoining ecosystems. Answer is A. The next one. In bacterial peptidoglycan, N-acetyl glucosamine and N-acetyl muramic acid are linked by, they are linked by beta-1,4 glycosidic linkages. Answer is option A. Then a mixture of para-dimethylaminobensaldehyde isoamyl alcohol and concentrated hydrochloric acid is popularly known as, it is called as Kovacs reagent. Okay. And it is used to detect indole producing microorganisms. All of the following are effects of water pollution with domestic sewage except the spread of infectious disease. Yes, domestic sewage can contain pathogens like bacteria, virus, certain parasites that can spread infectious disease. Increased eutrophication, yes, domestic sewage can also contain nutrients like nitrogen and phosphorus that can result in excessive plant growth and lead to eutrophication. Similarly, algal bloom also, because of the presence of excess nutrients in domestic sewage, it can result in increased algal growth or algal bloom. But decreased BOD is not a characteristic of increased domestic sewage. Because domestic sewage, it increases the pH of the water, because domestic sewage contains organic matter and the microorganisms break down by consuming oxygen. Break down these organic matter by consuming oxygen. Therefore, decreased BOD is not the answer, it is increases, or increased BOD is the answer. 5 cardinal signs of inflammation are, you must have read it as calor, dolor, rubor, tumor, functio, lisa. That means calor means heat, then dolor means pain, rubor means redness, tumor means swelling, and functio, lisa means loss of function. So the answer here is heat, pain, redness, swelling, loss of function. Answer is option A. To suppress the immune system and to prevent rejection after tissue or organ transplant, the drug commonly used is, so it is cyclosporine. So they have asked which is an immunosuppressant drug. That is why immunosuppressants is they suppress the immune system and prevent rejection of tissue or organ transplant. So an immunosuppressant drug here is cyclosporine. Answer is option C. Vancomycin, it is used to treat bacterial infection, bacitracin for skin and eye infection, and methicillin is treatment of bacterial infections. Then an inverted pyramid of biomass can be seen in, so it can be seen in ocean ecosystem. This is because the biomass of phytoplankton, that is the primary producer, is less than the biomass of the zooplankton, which is primary consumer, and this is in turn less than the biomass of the secondary consumer, that is fish. So it is an inverted pyramid of biomass in ocean ecosystem. Then next one. During apoptosis, which of the following phospholipid is exposed to the outer leaflet of the plasma membrane? So it is the phosphatidylserine which is exposed to the outer leaflet of the plasma membrane, and it acts as a signal so that the phagocytes can come and eat it or engulf it. That means engulf the cell and remove it from the body. Then match the following columns in the respect to gastrulation in frog. So ectoderm, it give rise to nervous system. So 1B. 1B is there in C and B. A and D we will not see. It is not 1B there. Then ectoderm, gut lining, 2A. So C is the right answer, because in B it is 2C, which is not correct. Mesoderm, skeleton, 3C. So 1B, 2A, 3C. Answer is option C. Then as an important part of the non-specific immune defense mechanism, which of the following enzyme is present in human tears, saliva and mucus? It is lysozyme. Answer is option C. It is involved in non-specific immune defense mechanism. Then during human menstrual cycle, progesterone is secreted by, it is by the corpus luteum. Answer is option C. Acetyl number refers to the number of dash required to neutralize the acetic acid liberated by hydrolysis of dash acetylated oil or fat. So acetyl number, it refers to the number of KOH required to neutralize the acetic acid liberated by hydrolysis of 1 kilogram of oil or 1 gram of acetylated oil or fat. Answer is option C. Then soil horizon layers are differentiated by, so they are differentiated by physical, chemical and biological characteristics. Answer is option A. Physical characteristics, they include the texture, structure, color, etc. Chemical characteristics, they include pH, moisture content, nutrient content, etc. And biological characteristics, they include the microorganisms that are present in the soil. Then next one. During mammalian early embryonic development, the trophoblast cells of the 16-celled morula develop into what is called as the chorion. Answer is option C. Then next one. Which of the following is not a secondary lymphoid organ? So there are two types, that is primary lymphoid organ and the secondary lymphoid organ. The primary lymphoid organ, they produce the lymphocytes, that is T cells and B cells. Secondary lymphoid organs, they maintain and activate the lymphocytes, that is secondary lymphoid organ. So primary lymphoid organs are your bone marrow and the thymus. Okay. So they have asked here which is not a secondary lymphoid organ. Secondary lymphoid organ, spleen is a secondary lymphoid organ, lymph node is a secondary lymphoid organ, tonsil is also a secondary lymphoid organ. Another secondary lymphoid organ is your mucosal membranes. Whereas thymus, I told you, it is a primary lymphoid organ. So the answer is option C. Then next one. According to the histone theory of Hanstein in 1868, the cortical part of the angiosperm leaf is developed from. So this is the shoot apex according to histogen theory. So there it is divided into three layers, that is dermatogen, peribulum and pleuron. The dermatogen, it give rise to the epidermis. Peribulum, it give rise to the cortex, and the pleuron give rise to the steely and vascular tissues. So that is it according to the histogen theory of Hanstein. So here they have asked the cortical part of angiosperm leaf. So cortex is derived from which histogen according to the histogen theory. So I told you it is from the peribulum as per the theory. Answer is option C. Then next one. One strand of double-stranded DNA is mutated, changing all the cytosines to uracils. After one round of replication of the mutated DNA strand, the Tm value of the resultant DNA will be. So what will be? The Tm means melting temperature. So this is your hypothetical DNA strand. This is DNA strand. It is having so many C and Gs. Now cytosines are converted to uracil upon mutation. So wherever cytosine come, I have put a U there in the first strand, because they have told only one strand of the double-stranded DNA is mutated. So this strand is mutated. The other strand I have left it as such. Now upon replication, this one strand, that is this, it will give rise to the next strand that is complementary in base pair to this. So wherever U comes, it will be an A, and this strand, it will give rise to the same strand as this because it is not mutated there. Now they have asked, after one round of replication, the mutated DNA strand, this one, what will be the Tm? Now it is having, since the mutated strand most of the bases are filled with A and T, that is mainly it is adenine and thymine or uracil, and they have only double bond between them. So it is easier to break the double bond than the triple bond between G and C. So when compared to this one, this will have a lower melting temperature. Okay. So the answer here is B, lower. Answer is option B. Then next one. In Arabidopsis embryogenesis, an apparent distinction between adaxial and abaxial tissues can be seen in. So it is in the torpedo stage. Answer is option C. Two mammals of India included in the red list of IUCN are. So they are the red panda and hoolock gibbon. Answer is option A. Then next one. The DNA polymerase involved in base excision repair is. So it is beta that is involved in base excision repair. That is, it will base excision repair. It will correct the damaged DNA bases, and it is DNA polymerase beta that is involved. Answer is option B. Histone acetylation increases transcription of gene because. So histone acetylation means it will add an acetyl group to the histone proteins or histone amino acids. As a result of this, it will loosen the bond between the histone protein and the DNA, and this will result in certain regions of the DNA getting accessible to the RNA polymerase. As a result, the transcription increases. So it increases DNA histone interaction. No, it reduces DNA histone interaction. The acetyl groups of histones are recognized by RNA polymerase. That is also not correct. It loosens the DNA histone complex, making it more accessible to RNA polymerase. That is correct. It increases DNA bending, which is recognized by RNA polymerase. No. So answer is option C. Then which of the following transposable element causes hybrid dysgenesis? So hybrid dysgenesis, it results in sterility or genetic abnormalities of the offsprings if there is a cross between different strains of the same species. Okay. Different strains of the same species upon crossing will give rise to offsprings that are sterile or have certain genetic abnormalities. That is called as hybrid dysgenesis. And one of the cause for hybrid dysgenesis is transposable elements. And it is commonly seen in Drosophila, and it is because of the transposable element called as P element. Okay. So the answer here is option C. Then, in a cladogram, any branch with more than 2 lineages indicate. So this is the branch. This is the cladogram. This is the branch. So any branch with more than 2 lineages. So here you got one lineage. Here you have got 1, 2, 3, 4 lineages. So that is called as polytomy. Answer is option B. Then coming to homoplasy. Now what is homoplasy? That is, it is a character that is shared by different species but not present in their ancestor. That is, if you see here, this is B. This is the ancestor of that particular species, and here also there is B. This is the ancestor of this B, but the B character is not present in this ancestor, which is it is A character that is present in the ancestor. So this species represent homoplasy. Okay, then what is parsimony? It is it is choosing the evolutionary tree from a group of evolutionary trees, and you choose the one which is having the least number of evolutionary events or least number of evolutionary changes. So you select that particular tree. That is called as parsimony. And simpliciomorphy. Sim means shared, and plecomorphy is that represents ancestral character. So simple isomorphy is it is common between two organisms present in multiple lineages. Okay, that is simpliciomorphy. So here the ancestor also will have the characteristic, and their great ancestor also will have this particular characteristic. That is simpliciomorphy. So this is a ancestral character that is present in the offspring. So it is simpliciomorphy. Then next one. In evolutionary studies, dolo parsimony refers to. It refers to an advanced state evolved only once. Answer is option A. Then clathrin coated vesicles mainly transport proteins from. So clathrin coated vesicles, they are involved in receptor mediated endocytosis, and they transport proteins from plasma membrane to endosomes, and it can be either for sorting or for degradation. So answer here is plasma membrane to endosomes. Answer is option C. Then anagenesis means. So anagenesis means there is a gradual evolution in a single species over time. Okay, and this does not involve splitting of the species and all. Okay, it results in the accumulation of these changes within the species, so that this species is getting converted to another species. So the answer here is gradual change that occurs in a species without the need for splitting. Answer is option C. Then next one. Choose the correct hierarchical sequence of the following geological periods or epochs. Ordovician, Pennsylvanian, Triassic, Cretaceous, Paleocene, Holocene. So the answer here is option A. So thank you for watching my channel. Please do like, share and subscribe. If you have still if you want still previous year question papers of PUMDET been solved, you can go to my channel Omni Bioentrance. In the playlist you can see for PUMDET folder, in that you will find. Thank you.